Hello, everybody. This is Miss Peachy from WCA Biology A class. And what I'm going to go through today with you is your second portfolio of the semester. It's actually your final portfolio for the semester. Believe it or not, you only have two portfolios for first semester here. So this is from Unit 5, Lesson 4, and this is your photosynthesis portfolio. Now, again, in this portfolio, they are going to want you to design and carry out your own experiment. So when we look at the course outline and we go to lesson 5, which is photosynthesis portfolio 2, because there's 3 lessons, one where they introduce the portfolio, one where they kind of go through more details of the portfolio, and one where you will turn in the portfolio. If you look at lesson 5 and you go down to, I believe it's slide, let me find it here. It is slide 6. So in slide 6, you will see a link for a photosynthesis portfolio lab worksheet. And when I open that up, you get this. Now, if you decide you want to actually do this hands-on, follow the directions that are found in the lesson, and carry out this experiment at home, which you totally can do, then this is the worksheet you will need to use. Do not type directly into these boxes. It looks so tempting, doesn't it? You can just type in here, but this will not save as is. You will need to first download it. You will need to save it to your computer and then open it again in your computer, put in your answers, and again, save it with all those answers in there, and then you can upload it to me in the Dropbox. If you just start typing in here and try to save it, it's just going to delete everything that's in your document. So do yourself a favor and first save, then alter, then save again, and submit it to me. If you say, I don't have these materials, Miss Peachy, then we'll be doing a different kind of video portfolio here at the end of this recording here. So just stay tuned. We're going to kind of go through it, and I'll give you something that you can use and take data from. If you're planning on completing the one in the lesson, then you would go to Unit 5, Lesson 5, page 7. In page 7, it goes through exactly what you need to do for this portfolio. So you're going to need some materials. You're going to need bicarbonate solution, that's just baking soda. You're going to be needing a couple of plastic syringes. You are going to be needing some spinach leaves. You are going to be needing some clear containers where you'll be able to place your leaf discs and leave them in bright light to make observations. I'm not going to go through that portfolio with you here because the directions are right included. They're pretty easy to follow, and if you choose to go that route, like I said, that's awesome. Go ahead and do that. You have that option. It's nice to be able to do them hands-on, but this is primarily for students that can't do these hands-on for one reason or another. They don't have the materials. Maybe they don't, they just aren't able to accomplish that. So let's do a different thing, and we'll do it on the video here, and I'll kind of explain to you what you're going to be doing because there's a little bit of a change. First of all, the lab worksheet that you are going to be used is a little different. It's very similar. I kind of took the first one and just modified it a little bit. You'll find this lab worksheet in the link underneath the video here. And there's a few differences. The first difference is a modification to this first data table where I added a column that says constants or a row that says constants. So you're going to identify what your control is, the independent and dependent variables, and the constants. Now I'm going to tell you in this situation, you're going to have multiple controls and ultimately multiple variables that you're testing. You're actually testing 2 variables, so there will be 2 independent variables as well, 1 dependent variable, and then a multitude of constants. Remember, constants are things that you don't change. You keep them the same from one trial to the next so that we know they're not affecting the outcome. And then you're going to describe the materials that were used in the video. Please don't just say, I used a video. No. Describe the materials that are used in the video. So you're describing these materials and the procedure as though you were actually doing it yourself, okay? And then finally, what we're going to be looking at is just observations of 4 different vials, vial A, B, C, and D. You're going to make observations of that at the beginning and then after 12 hours. And then lastly, we're going to analyze that data. You are going to determine, you're going to explain how you can determine from those results whether or not photosynthesis took place. And then you're going to describe an additional lab investigation you might be able to perform using the same technique if you wanted to investigate further. Like what other things might impact photosynthesis and how could I test them using this lab technique? All right, so this is the data sheet you're going to complete. And notice that is because it's a Google Doc. If you're not familiar with Google Docs, you have a couple options. You can go to File and make a copy, and you can make a copy of this document. You can edit your copy, and then when you're done with it, you would go to Download as a Microsoft Word file to your computer. It would save to your computer, and you could upload that to the Dropbox. Or you can just right away go to File, download as Microsoft Word, and then make your edits in Microsoft Word and save it and upload it to the Dropbox. But ultimately, that file that you upload to the Dropbox, it should be a Word document because that's all the ones I can open using the Dropbox. All right, so let's go ahead and look at this video. I'm going to explain it to you. I'm just showing you the video part of it. I'm going to mute the sound because that's going to be me that explains it to you. And then we're going to kind of see how that applies to what's going on in your lab worksheet. I'm going to go ahead and bring this over. This is actually on YouTube. Things go a little slowly sometimes when I do this. So I want you to notice right off the bat, I'm going to actually click and drag, move this toolbar out of the way. I want you to notice right off the bat that we have 4 tubes here, 4 vials. That's either V-I-A-L-S or P-H-I-A-L-S. That's how you spell it. But so we have 4 vials labeled A, B, C, and D, and then they're kind of a little bit of a yellowish green in color, right? So what has been placed in here? This is just water, and this is water with an indicator solution that indicates for the presence of carbon dioxide. The indicator solution, when carbon dioxide is present, will be yellow with high concentrations of CO2, green with medium to low concentrations of CO2, and blue when there's no CO2 in there whatsoever. So as you can see, we have all 4 of them the same color, just this light yellow, kind of a greenish yellow color. This would be the observations you would make at time equals 0. So if you look at your data table, this is going to be your first observations. Okay. So I'm pushing play on here. It's probably where they explain exactly what I just did. And now we're going to place plants in vials A and vials C. This plant is called Elodea. Elodea is an aquatic plant, so it likes to live in water, and it does photosynthesis in the water. And so we're going to place 2 of our vials that have plants in them and 2 without. So now think about those variables, okay? So we are going to be testing if a plant in sunlight, if we can see evidence of photosynthesis, or which of these situations would we see evidence of photosynthesis? I want you to think about photosynthesis for a moment as well. Photosynthesis takes carbon dioxide and it uses that carbon dioxide in water in the presence of sunlight to make sugars and oxygen, right? So at this point, we have all 4 vials that have a lot of photosynthesis in them. So we'll see what happens next, and then I want you to make a prediction. All right, so we have our 4 vials. So let's see what the different colors mean. That's kind of what they're going to talk about next here. Talk about what this light yellow actually means here and what colors we can expect to see. So light yellow tells us high concentrations of CO2. Green tells us low concentrations, and blue would be none, right? So what we're going to do here is we are going to place, let me see if I can pause this for a moment, but we are going to place, oops, shoot. Let's go back to that spot because here we go. All right, sorry about that. So we are going to be placing caps on these, and we are going to place 2 of them in sunlight. And then they're going to take the other 2, actually using a bright light like an incandescent light, and they're going to wrap them in tin foil to try to block out the light. So let's just pause and take inventory of what's going on here, right? So ultimately, we're looking at 2 groups, our test group and our control group, but we're also testing 2 variables. So our variables that we're testing is, do plants in bright sunlight produce, well, we're testing 2 variables here. We're testing one with a plant and one without. And so think about again what's happening. We are measuring carbon dioxide in the water. So I want you to think about your expectations. Make a prediction. We're going to spend 12 hours here just letting this process happen. And remember, yellow is high concentration of CO2. Green is medium or low concentration of CO2, and blue means no CO2 at all. Photosynthesis, when it occurs, CO2 is removed and used to make sugar. So what colors might you predict these solutions to be after 12 hours? So pause that, make your prediction, your hypothesis here. Which of these vials might change color, if any, and what colors would you expect to see after the 12-hour window has passed? 2 in the dark, 1 with a plant and 1 without. 2 in the light, 1 with a plant and 1 without. So again, pause, think, write down your hypothesis. Okay, so hopefully now you've paused. Let's go ahead and see what happens here. 12 hours have passed, and we're unwrapping them. And now let's look at what they look like. And this is your observations you would write in the data table in the second row. Note the colors of the solutions themselves. Don't be like fooled by the fact that there's a green plant in there, but just look at the colors of the solutions themselves. Does this fit with what you predicted would happen? Again, remember, yellow means high concentrations of CO2, green would mean low, and blue would mean none. How can you explain the results based on photosynthesis? Or how do the results show you that photosynthesis occurred? And in which vial or vials would you say photosynthesis took place? Okay, so after you're done making all of your observations and kind of determining the results of this experiment, I want you to go back to our photosynthesis portfolio lab worksheet. I want you to answer these questions. How can you determine if photosynthesis occurred based on your observations? Describe an additional lab investigation you might be able to perform about photosynthesis using the same technique. Okay, so think about it. How would you know, for example, how quickly photosynthesis happened? How might you know, how might you alter some of the things you're testing, like the amount of light? And will that have an effect on photosynthesis? How would you measure that? How would you measure if different plants you're introducing? Think about all the things that you could change to still kind of do the same procedure, but measure a different aspect of photosynthesis. And then relate your conclusions to your hypothesis. Were you correct in your prediction? And reflection, what would you change if you completed another trial? How would you revise your hypothesis if you completed another trial of the experiment? So all of those kind of conclusion questions to answer. The data collection, just do from the video that we showed. Again, if you want to do the original experiment, you are totally welcome to do so. But if not, then this is a good supplemental. If you guys have any questions, let me know. Otherwise, good luck, and we'll see you in the next video.